Okay, so let's restart that introduction again. Thank you everybody for joining us for the 5th annual 3-minute pieces competition. I know at this point we've been doing this online thing for over a year, so I very much appreciate you folks keeping through and still joining us over Zoom. So with us we have 3 judges today. So Dean Cool, so Dean of the Guarini School. Dean Cool, I wasn't able to find you. Also graduated from Dartmouth as an undergrad, correct?
Yeah, class of 1988.
Okay, 1988. I'll definitely update it so we can have these files archived. And then we also have Barry from the Tech Transfer Office.
Professor Call's a youngster compared to me.
And then we also have Sadie, alumni and currently a project manager at Simbex.
Hi, everyone.
And I guess we can get started. So we'll have a hard stop of 3 minutes for each contestant. I'll try to message you folks individually when you have 30 seconds left. But I'll stop sharing after I announce, and then once you start, it starts counting. So don't feel like I'm eating into your time when I announce your name. So first up, we have Rachel on work-hardening of metallic alloys.
Wait, sorry, are you going to tell me go?
Yeah, as soon as you start, I'll start counting, but
okay. All right, so my name's Rachel. I'm a 4th-year PhD student at Thayer. I work with Professor Ian Baker, and I study metallurgy. So in high school, probably like a lot of you, I was taught that metals are just like a sea of electrons just hanging out. And that's not true, but they're also crystals, which is much more exciting to me personally. And so the metals I study specifically are binary intermetallics. And it's been known for decades that these have this weird property where when the crystal is disordered, it's not very strong. And when the crystal is ordered, the strength increases by about 50%. And this is really great. We'd like to do that to other metals as well. But nobody's really figured out why exactly this happened. So my project is focused on the basic science of this phenomenon and trying to figure out in a quantifiable way why intermetallic alloys work harden so much more. So I mentioned ordered versus disordered crystals. The metals I work with are face-centered cubic. So their crystal structure is literally like a cube with atoms on the corners and atoms on the faces. And when it's disordered, the atoms just sit wherever they want. So I work with nickel-3 iron, and they'll be wherever they want. And then when it's ordered, the nickel atoms sit on the face sites and the iron atoms sit on the corner sites. And this seems like a really small change, but as I mentioned, it increases the strength by about 50%. So the main thing that people think contributes to this is a little defect called an antiphase boundary tube. And they roughly look like parallel pipets in the material. It's a form of chemical disorder that's a few nanometers thick and many microns long. And so the crux of my research is to introduce these defects into the material and then get rid of them via heat treatments and then see how the material changes. So the main techniques I use to study this are tensile testing, which is how we get the strength by pulling the metal apart, as well as magnetization studies. So seeing how the magnetic properties change with the deformation and annealing, because nickel-3 iron is also a soft magnet, and also differential scanning calorimetry. So we can see the heat flow that indicates when reactions are occurring in the metal. And actually, so far, most of my tests have shown that antiphase boundary tubes don't do anything, which is a little disappointing as a student, but could be really interesting to prove this theory that's been around since like the 80s might not actually be what's happening, at least in this specific metal. So moving forward, I'm going to focus a bit more on the microscopy side of my research and watch the metals deform in real time and see if I can figure out what is actually causing this extra strengthening effect. Because as I said at the beginning, if we actually understand why it's happening, we can use it to engineer better metals beyond these binary intermetallics, which honestly aren't actually that useful for anything other than basic science. But for basic science, they're great. And I hope my thesis will expand the science behind them to be useful to things that actually have engineering applications. And I'll wrap up a little early, so thanks for your attention.
Beautiful work, beautiful, beautiful. Okay, so those were 3 minutes, and then we can move on to our next speaker. Oh, oops, I forget that the thing actually goes off. Okay, so I feel like I'm gonna take more time trying to reshare my screen than what people take talking. Okay, there. Okay, and so for our second speaker, we have Al. And they'll be presenting on 21st Century Poetic Conversation with Emily Dickinson on the matter of Black lives.
All right, let me know. Am I good? Can you guys see me and hear me? Okay, excellent. All right. The lamp burns sure within, though serfs supply the oil. It matters not the busy wick at her phosphoric toil. The slave forgets to fill, the lamp burns golden on, unconscious that the oil is out, as that the slave is gone. My name is Salehi, and for my Mal's thesis, I am writing a compilation of original poetry in conversation with Emily Dickinson regarding Black Lives Matter in the 21st century. You see, the CERN recently observed tachyons are capable of moving back and forth in time, and there are theories about wormholes that can move matter backward in space-time. It was also observed in the blockbuster movie Interstellar. I want to be replying to her posts via Interstellargram, chatting through poetry and vice versa. You see, she was a futurist with poems that were waiting to be published at a later date in our time. And she writes about the slave inevitably getting away. I want to write poems and continue that conversation and excavate the meaning for today. She lived at a time when we questioned whether Black lives were merely chattel or were they entitled to inalienable rights as human beings under the Constitution. Although 150 years or more have passed, her work is timely and eternal, and I believe it mirrors my own work. Asks, how do we value life? And the question of worth is still being debated today. Does she still matter on the question of Black Lives Matter? The Emily Dickinson Museum seems to agree that she does. What does she have to offer? What resources does she have to shed light on that subject? She was viewed differently at her time as a crazy woman, yet she apparently now, in recontextualization, she was very much connected to those timely questions. My project aims to also deal with these ontological questions, to deal with the timely as well as the eternal. I want to tell her that things do get better, but unfortunately they are far from perfect. And ultimately, I hope that 150 years from now, another poet can have the opportunity to reply to the two of us and say that, in fact, this issue has finally been put to rest. Thank you for listening.
Thank you so much. That was great. Yeah, appreciate you folks, all of you a bunch. So next up, okay, I think this works a little better. Okay, we have Catherine, and she'll be talking on evaluating the accessibility and accuracy of obesity-related health information. And you can start, there we go, perfect.
The COVID-19 pandemic has amplified our access to information, including misinformation or content that is inconsistent with scientific consensus. COVID-19 misinformation is multifaceted and can feature various subtopics, including on associated risk factors. One of the most prominent risk factors for COVID-19 is obesity, which is also a comorbidity for other chronic and non-chronic diseases. As obesity prevalence continues to increase, it's imperative that we evaluate both the accessibility and accuracy of obesity-related information from those around us, including physicians, friends, and strangers. And it is this need that sets the foundation for my dissertation work. My first project focuses on physicians who specialize in obesity medicine. Using geospatial and mathematical modeling, I've evaluated both physical access as well as the practicality of access. In other words, does the supply of physicians meet patient demand? Along the way, I've developed tools to effectively, affordably, and rapidly collect physician-level characteristics from online materials, foregoing the need for expensive data sets. My second project looks at how friends discuss obesity. Using data from Facebook and Instagram, I'm leveraging machine learning and natural language processing to identify how distinct conversation topics on obesity have evolved throughout the COVID-19 pandemic. Knowledge of the distribution and diffusion of these topics could aid in the development of educational materials or other interventions to mitigate the spread of harmful obesity-related content. My final project looks at how strangers discuss obesity. Using 9 years of Reddit data, I've again turned to machine learning and natural language processing, this time to identify whether there are distinct linguistic and semantic features that differentiate misinformation, weight-related stigma, and factual content. The identification of these features could aid in the development of tools that automatically detect and remove harmful content from the platforms, instead of the manual time-intensive tagging that we currently do. As technologies continue to evolve, it is imperative that we identify barriers to quality health information and develop tools to overcome them. While my work centers on obesity, the tools have been developed in such a way that they can be applied across domains. By elevating our access to trusted officials like physicians and reducing our access to harmful content, such as misinformation, we can end this infodemic once and for all. Thank you.
Thank you very much. So next up, okay, let's see. Oh, there we go. Okay, so I think it's Swetha, but can you let me know how to actually say your name properly?
Yeah, that's fine. It's Swetha.
Okay, Swetha. Okay,
yeah,
perfect.
Thank you.
And so the talk is titled Social Life of Microbes.
Okay. Hi, everyone. I'm Swetha, and today I'm going to talk to you a little bit about the social life of microbes. Microbes are tiny living things that cannot be seen by the naked eye, so you need a microscope, but they're present everywhere. They're present in air, water, soil, they're present on us, and they're also present inside us. Microbes also help us run the world. They're important in big scale processes like making nitrogen. They're very useful to us. They help us make things like medicine. They help our crops grow better. Microbes, unfortunately, also cause diseases. Interestingly, microbes, like humans, can live in communities. And depending on the kind of microbes that are present, they behave differently, ultimately affecting the overall process. Microbes can help each other, they can fight with each other and go to war, as well as they can work together. And sometimes when microbes are working together, there are cheater microbes that like to do none of the work and take all the advantage. But there are also policing techniques that microbes can use to ensure there's no cheating. For my project, I'm looking at interaction and how this would affect disease progression. So I'm studying a bacteria and a fungus that interact in a disease called cystic fibrosis. Patients with this disease end up having a lot of microbes grow in their lungs. And this affects their disease and how bad it can get. So although, like I mentioned, there are many microbes that grow, I'm using one fungus and one bacteria and using a special kind of microscopy called confocal microscopy, where I can take three-dimensional images of these microbes, and then we can locate how they are behaving in space and infer interactions from that. What I found was that these microbes behave very differently in communities versus when they grow alone. We also saw that a community of the fungus and bacteria together is bigger and taller than a community of, say, just the bacteria or just the fungus. We also saw that the bacteria is attaching to the fungus, implying that there's some interaction that needs contact. I'm still studying what the mechanism of action is here, but studying these interactions and how microbes behave with each other can help inform us about how a disease is progressing, and more importantly, can help us find better solutions and medical techniques to combat them. So understanding how microbes behave and the social life of microbes will help revolutionize our thinking about the big scale processes. That's all I have for you. Thank you.
Thank you very much. Okay, so next up we have Billy Jin, and with a talk title, Energetic Heart.
Yes, it's my turn. Good evening, everybody. My name is Billy Jin from Thayer. I'd like to start my talk with a couple of questions for you guys. Do you know what the leading cause of death is in the United States last year? Let me tell you, even with the COVID-19 pandemic that caused over 500,000 deaths, heart disease is still the #1 killer. Do you know how many cardiac pacemakers are implanted every year in the US? It's about as many as 200,000. However, Those devices are run by batteries, and batteries do die sooner or later, depending on how your heart depends on it. Usually, the battery in pacemaker can only last for 5 to 7 years. So, a patient may need to receive several replacement surgeries after the first installation. You know, this replacement surgery will bring about tremendous burdens and the risks to the patient. Can we avoid these extra surgeries? The answer is yes. For my thesis work, we developed various energy harvesters based on piezoelectric materials. These energy harvesters can collect the mechanical energy from the heart beating and convert them into electrical energy to charge the battery of the pacemaker, and therefore significantly increase the lifespan of the battery. In designing the energy harvester, we focused on a few important things. First, energy generation. We optimize the material's performance. We use biocompatible energy materials. Nanotechnology is used to engineer the structure and composition of the material. Second, flexibility. Since the device will have to directly contact with the heart soft tissue, it has to be flexible. In addition to using flexible materials, we use innovative structures to create ultra-flexible and stretchable devices. Next, implantation. We don't want to add extra surgical procedures to implant our devices. Therefore, we designed our energy harvester in a way that it can be incorporated into the existing commercial pacemakers. Lastly, clinical trials. In order to prove our device's feasibility and demonstrate its performance, we carried out our test with animals. In the experiment, surgeons can easily implant our device on the pacemaker into the right ventricle of the heart. The energy harvester can generate over 3 volts voltage and the maximum power density of 40 micron watts per centimeter cubic. That meets the power requirement of a commercial pacemaker. So currently, we're working on long-term clinical tests, and we're very, very excited to see the result. Thank you, everyone.
Thank you very much. I also just want to congratulate everybody for the wonderful talks and then being on time. I thought it was going to be an issue where I had to call people out, and it's been wonderful. If you're one of the next speakers and goes over, I'll still be nice about it, I promise. Okay, so next up, we have David with a talk titled, "Oh, the Places You'll Snow, Studying Snow on the Arctic Sea Ice."
Awesome. Thank you, Alberto. Hi, everyone. My name's David. I wish I had the talent to deliver this in verse like Dr. Seuss and Salehi, but I don't. Everyone's probably enjoying the spring right now, but I want to take you back a few months to think about snow. And actually, I don't want you to think about so much the snow here in Hanover, but I want to think about the snow up on the sea ice in the Arctic. Now, you might not think about Arctic sea ice very much, but it's actually very important to our world and to our climate. First off, think about how large it is. Every winter, an area roughly the size of the United States freezes and goes from what appears from open ocean to what appears to be solid land. Now, the ice freezes every winter, and in the summer, this ice acts kind of like a hat sitting on top of the earth that helps reflect sunlight away from it. And so having the sea ice up there is really important to keeping our planet cool. Now, as many of you may have seen, Arctic sea ice has declined dramatically in recent years. And so the amount of protection it's providing has decreased year over year. We want to be able to predict how much the sea ice is going to change going forward into the future. And it turns out the most important thing for figuring out how much sea ice you're going to have every year is actually how much snow you have on top of the sea ice in the winter. Now, in the winter, snow is an incredible natural insulator. If you ever get caught out here in New Hampshire, Vermont in the winter, you need to spend a night outside, you'll be much better off if you dig yourself a snow cave and sleep out in it, rather than sleeping out on top of the snow. Snow's like a blanket on top of the sea ice. and that blanket insulates the ice and prevents it from growing. And so one of the things we care about is how much snow is there on the sea ice and how is it distributed? Now, if you take your blanket and you just bunch it up on top of your feet, it's not going to insulate you as well as the blanket that you spread out over yourself. And snow on sea ice works exactly the same way. Sometimes it all bunches up and sometimes it doesn't. And my research is focused on determining under what conditions does it all bunch up, under what conditions does it spread out, and how might these change in the future so that we know how the future of the snow blanket on the sea ice will affect sea ice going forward, and how that will affect our climate. Thank you very much for listening.
Thank you very much. Okay, so for our next speaker, we have Harsha, and with a talk titled Surgical Sensing.
Hey guys, I'm Harsha. And if you think about it, in your lifetime, you'll undergo 1 or 2 surgical procedures due to various reasons, such as going to the dentist's office or, God forbid, having a tumor. So in those surgeries, the surgeon, if he or she or they are not assisted by an optical technology like an O-arm, which provides intraoperative X-ray, or have advanced facilities like what we have at the Dartmouth Hitchcock Hospital, where you can take intraoperative CT, they're often going blind based on their intuition and their understanding of preoperative images and their understanding of anatomy. So going blind into surgery where you're working with critical anatomy like vessels, nerves, it's dangerous and it may lead to chronic or acute conditions. One such application is dentistry, where they place implants to replace fallen off teeth or rotten teeth, where they drill through your upper and lower jaw, and more often than not, unless it's instrument guided surgery, they don't have an idea of where the inferior alveolar nerve lies, or where the sycardian membrane, which separates your sinus, lies. And about 50% of cases, they often nick it or perforate it, which causes like temporary issues like loss of sensation, severe pain, or chronic issues such as getting infected and losing actually your sensation permanently. And in order to avoid that, or in order to provide surgeon a feedback, we are exploiting electrical impedance properties of tissues. So imagine tissues like as cells with electrolyte in them, and if we pass this very small amount of current, it impedes the flow of current to a certain extent, and different tissue types impedes the flow of current in different ways. We design sensors to exploit that property of tissue and give surgeon an input when he when he's drilling real time to prevent he or she from like hitting the nerve or the membrane, or even like making sure all the tumors removed. By changing the properties, by changing the geometric properties, by changing the area, and by changing the element distribution, my research focuses on constructing these complex sensors that provide simple inputs to surgeon, such as go or stop. That's my research. Thank you for listening.
Thank you very much. Okay, so next up we have Abigail, or I don't know how to say that too well in English. Abigail? No. Sorry. So, and then the name of the talk is Brain Behavior and Herpes? Question.
Thank you, Alberto. We have learned a lot over the past 12 months. We have learned how to wear a mask in public spaces, how to entertain ourselves with TikTok challenges and charcuterie boards, and perhaps most importantly, we have learned how a virus can influence our behavior. Of course, most of the behaviors I just described were cultural phenomenon caused by fear of contracting illness and spreading disease rather than viral infection itself. But recent evidence suggests that certain viral infections may also be capable of causing permanent changes to behavior in the absence of social pressures. Popular news media has reported on COVID brain, describing a group of patients who have recovered from SARS-CoV-2 infection, but continue to experience deficits in mental faculties such as sustained attention and concentration. And at the same time, my lab demonstrated how herpes simplex virus may also have long-lasting effects on the behavior of infected mice. Now, we've long understood that our behavior is a product of our nature and our nurture, but we do not typically think that our behavior is a product of viral infection. Furthermore, when we think of herpes, we are taught to think of cold sores and sexually transmitted infections, not the ways in which we interact with the world. Understanding how viral infection affects the brain may be the key to understanding how viruses affect behavior. For my thesis, I aim to study neonatal herpes simplex 1 infection and its importance on affecting the brain and behavior of mice months after they were exposed to the virus. I hypothesize that the developing brain is particularly susceptible to viral infection and that neonatal herpes infection may cause persistent changes to developing neuroanatomy, neurophysiology, and learning and memory. To test these beliefs, I first plan to use behavioral tasks to assess learning and memory in 3 to 5 month old mice who were infected with low doses of herpes virus as neonates. There's literature to suggest that Alzheimer's disease may be linked to herpes infection. Understanding the way in which infection occurred at such a vulnerable developmental time point immediately post-birth contributes to later changes in learning and memory may help to elucidate this connection between neurodegeneration and viral illness. Next, I plan to stain and record activity from both mouse and human neural tissue to assess neonatal herpes impact on the developing brain. By using both mice and cerebral organoids grown from human stem cells, I will be better able to make predictions about how herpes affects the human brain and see how my mouse model compares. Herpes simplex virus is not nearly as scary as COVID-19. However, with an incidence of over 65% globally, it is extremely important to understand the impact of this seemingly common disease. A tiny virus may have enormous implications for infected neonates, and I aim to spend the next few years of graduate school investigating not just the often connected brain and behavior, but brain, behavior, and herpes viral infection. Thank you.
Thank you very much. And our next speaker is Anne. And the slide, okay. With a talk titled The Evolution of Optimum Growth Temperature Extremophilic Bacteria.
So my thesis deals with investigating how evolution progressed in creating the temperature optima in extremophilic bacteria. If you look for life in the most extreme environments on the Earth, you typically find it, despite what we thought many, many years ago. You can find microbes living in the Arctic Sea, and you can find microbes living in the hottest deep sea vents. So life has this incredibly wide range of temperatures, and it's an incredibly diverse range in which it can survive. But if you select any specific species, that species has an optimum growth temperature and a very narrow range around that optimum growth temperature in which it can live. So how did we evolve this incredible diversity across the tree of life, even though every sort of individual has a really narrow range? So to sort of investigate how evolution progressed to develop these optima, I look at a phylum of bacteria called the Thermatogoda. These are hyperthermophilic, thermophilic, and mesophilic bacteria that we have isolated from deep sea vents and oil wells and other subsurface environments. And they're all a monophyletic clade, so they're all related, closely related. We can call them sister taxa. And they all descend from a common ancestor. And you might expect that a bunch of things that have descended from a common ancestor would share a bunch of similar traits, just like you and I share similar traits with our brothers and our sisters from our parents. But in this phylum, temperature is not one of those shared traits. The species in this phylum have a huge range of optimum growth temperatures. Some of them are mesophiles, surviving at temperatures below 50 degrees Celsius, all the way up to hyperthermophiles, which survive at temperatures above 80 degrees Celsius. So this makes a great study system for trying to understand how different kinds of evolutionary processes can drive shifts in temperature. And I used a couple of different tools. One of them is phylogenetics. So phylogenetics is going to, it sort of helps us understand the relationships between the organisms. and the relationships between the genes that compose the organism's genomes. Phylogenetics allows us to make best inferences about the temperature of the last common ancestor, so we can then make hypotheses about how temperature has shifted over time. And phylogenetics also allows us to identify genes that are not shared from the parents. Perhaps they were gained from an outside taxa, like a friend or something. So phylogenetics can show us how horizontal gene transfer might impact temperature evolution. And second, I use comparative genomics to look at gene presence absence and genome composition similarities and sequence differences between taxa to better understand how gradual evolution may have impacted temperature optima in these microbes. Thanks.
Thank you. And then our 10th speaker is Sumi, and with a talk titled Repositioning FDA-approved drugs to target cancer stem cells in glioblastoma.
Thank you, Alberto. All right. Hi, I'm Sumi. I'm a second-year student in the MSB department. So pretty much all of us in our lifetime will encounter at least one person who is battling with cancer. And this is a sad reality because close to 50% of the human population has some sort of benign or malignant tumor. Brain tumors specifically are extremely debilitating because the nervous system is so fragile and essential that any shift from normal has long-lasting effects, even if the tumor is benign. In our lab, we specifically studied glioblastoma, which is a late-stage brain tumor, which has one of the lowest patient survival rate post-diagnosis. The problem with current cancer therapies is that they're almost never able to prevent recurrence of tumor. It is believed that this could be because of tumor heterogeneity, which essentially means that there are many subpopulations of cells within a tumor that are different enough that any one type of treatment cannot kill all the different populations. One such subpopulation is the cancer stem cell-like population. These cells are similar to the cells we see during nervous system development and are possibly the root of tumor growth and recurrence. So we are studying oligodendrocyte precursor cell, or OPCs, which is a type of stem cell which during development normally grows into adult oligodendrocytes, which is a type of nervous system cell. In the adult brain, however, these OPCs are nurtured by the rich neural microenvironment that has high blood supply and nurturing chemicals like neurotransmitters, one of them being acetylcholine. So acetylcholine during development helps these OPCs divide and grow. And the OPC model that I'm studying highly expresses receptors to acetylcholine. And it was also noticed that glioblastoma tend to mostly be located in regions of the brain that have high level of acetylcholine signaling. So I decided to better understand this cholinergic signaling pathway in the OPCs and try to obstruct it by using anticholinergic drugs. The best part about studying the acetylcholine signaling is that we already have a variety of different anticholinergic drugs that are FDA approved and cross the blood-brain barrier, which honestly is one of the biggest difficulty in developing any drug for any issue in the brain, which is actually getting it into the brain. So that problem is already solved. The anticholinergic drug that I'm using is benztropine. So can we actually eradicate cancer? Possibly in time. For now, we're trying to combine current cancer treatments like chemotherapy and radiotherapy with cancer stem cell targeted therapy so we can affect a wider population of cancer cells and improve patient survival rate. Thank you.
Thank you very much. Okay, last but not least, our 11th speaker is Vishal with a talk titled, I'm gonna try my best on this one. Okay. Metabolome analysis of Clostridium thermocellum at high substrate loading for efficient ethanol production.
Thank you. I'm second-year PhD student at Lean Lab, and our lab mainly focuses on the production of biofuels. So let's talk about global warming, one of the most pressing issues of the current world. So along with global warming, the population is increasing rapidly. So with the increasing population, the effect of global warming is sure to increase. And so one of the major contributors for this global warming is the extensive use of fossil fuel. So there's an urgent need for the alternative to fossil fuels. So one of the most abundant alternative sources is sunlight itself, but it is limited by large scale storage problem and the less efficiency, around 90% of the solar panels. And next is the use of electric vehicles, but which will be limited by the need of replacement of battery and their proper disposal. So biofuel can be one of the best alternative to the fossil fuels, as it is renewable, sustainable, and carbon neutral. So in our lab, we use Clostridium thermocelum, which is an anaerobic thermophilic microbe, and it can naturally utilize cellulose to produce the biofuels. It has been subjected to numerous engineering technologies by prevention of the formation of byproducts and introduction of genes from different microbes, but with limited success. The ethanol production has been limited around 30 grams per liter. So for the commercially viable production, we need at least 40 grams per liter ethanol being produced, and the substrate loading should be at least around 100 grams per liter cellulose or like biomass. So our main aim is to understand why growth and fermentation steps at high substrate loading, let's say 50 grams per liter cellulose or above. So we aim to fulfill this by understanding the change in intracellular and extracellular metabolites throughout the fermentation. So along the way, what we found out was, like, first was the accumulation of glucose. So we deleted the gene that was responsible for repressing the glucose uptake by the microbe. So again, then comes the pH limitation problem. So we solved this by just switching to pH controlled bioreactor. And the most important factor during the extraction of metabolite is the filtration time, because some of the metabolites, they degrade within fraction of seconds. So due to the viscous nature of the culture, the filtration was inhibited. So we tested almost every types of filters that are available to us, like nylon, polycarbonate, mixed cellulose ester, glass fiber, and everything. So with that, we chose the filter that has fastest filtration with maximum cell retention. So based on our preliminary data, we see a kind of bottleneck on the upper glycolysis of the central metabolism. So by further corroborating this data, our next strategy will be to generate an engineering technique that will relax this bottleneck and develop the strain for commercially viable ethanol production. Thank you.
Okay, so that's the end of our speakers, and in a lot of ways, I guess the end of the event itself. I just want to thank everybody for coming out. We had a pretty good showing. I messed up a little bit on the crowd choice poll, so I have to email that out. But then the idea is to announce the winners and then the crowd favorite, and then also publish the video at the same time for anybody who might have not been able to make it.